So yes, thank you for having me, and thank you, Professor Gleeson, for the excellent talk. So as Shonit said, I'm going to be talking about prostate MRI and about how AI may be used in improving the diagnosis, as well as how the current limitations and issues we're seeing, and how we may be able to overcome that with an AI challenge in conjunction with the NCITA adopted study, ReIMAGINE risk. So as Professor Caroline Moore's already touched on, the current pathway for prostate cancer diagnosis is following a raised PSA blood test. They would undergo an MRI, and a radiologist would find suspicious lesions, and this is an example of one, and they would score that lesion on a scale between 1 to 5 on how suspicious it was. If they found these lesions, they would then go on to have targeted biopsies to prove if they did or did not have cancer. And this graph just shows the proportion of these scores from 1 to 5 for which do not contain significant cancer. And you can see that as the score gets higher, then the more likely there is cancer. And those of low scores of 1 to 2 can safely avoid biopsy, but we routinely biopsy those of 3, 4s, and 5s. Particularly for those indeterminate 3s and 4s, you see a lot of unnecessary biopsies. So about half of the biopsies we do end up being negative for cancer. So it would be great to reduce these unnecessary biopsies, as there are risks involved with biopsies. So AI may be one way of kind of improving that or reducing those unnecessary biopsies, as well as other more simple biomarkers such as PSA density. But just to kind of simply show you what an AI may do is, again, as usual, radiologists would look at a scan, they'd identify an area of suspicion, they might score that lesion, and if it was a 3, 4, or 5, and they were going to have a biopsy, they might draw around that lesion, have some additional clinical data, blood tests, etc., input that into an AI algorithm of some sort, which of course there are many kinds, and that could give you a kind of risk for cancer, which you could either take into account or threshold to kind of aid in your biopsy decision for each patient. So hopefully that may, in theory, be able to reduce the number of unnecessary biopsies. There are lots of people already trying this. So we did a systematic review in the literature to see how close any of these algorithms were appearing to perform well, and if they could be used in the clinic, and what was maybe stopping them from being adopted. And although a lot of these studies quote saying there's promising results which match or even exceed radiologists, there were a number of shortfalls that we observed, and that's particularly in biased patient cohorts that we use for training and testing. There was no prospective studies being done, as Professor Gleeson mentioned, a lot of retrospective data. The big thing was a lack of external validation. So that means testing these algorithms on data which is different to what it was trained on. So an idea of, can it be generalizable to new centers, new scanners, etc. And so often they were single center, single scanner data sets. The outcome measures they chose were often quite poor and clinically irrelevant. And the test sets were either they didn't show any kind of power calculations, but often they were underpowered for the conclusions that they made. So hopefully this study, ReIMAGINE Risk, can help address some of those issues. So this is an NCITA adopted study, which is a cohort study which is looking at deeply phenotyping patients who have suspicious MRIs and go on to have biopsies. So we've got just over 1,000 men in ReIMAGINE Risk, all of whom have these contoured MP-MRIs, which you can see on the left, targeted biopsies of all their lesions, additional blurred urine and tissue samples for further biomarker development. And so we're gathering a lot of information about these patients, and as you can imagine, they'll be very useful for developing and testing AI. So we've also, I should also mention, as part of ReIMAGINE, there is a consortium of both academic and commercial partners who have expertise in a number of different areas, including radiomics and AI in imaging. And we're kind of working with them to develop novel biomarkers. So one of the things we're doing is this AI challenge, and what that means is we're sending a small subset of the data as a calibration set to a number of these AI partners, and we're giving them some time to develop an algorithm which we will then test ourselves based at UCL 6 months down the line. And so what that allows us to do is to a priori set what we think is a clinically relevant performance and task and metrics that we want these algorithms to hit, that we can evaluate them independently from the companies. We have an appropriately powered large test set of about 800 participants, which is external and has multiple centers and tens of scanners from all major vendors. We can also make comparisons against more simple other clinical biomarkers such as PSA density, and also between algorithms. And the hope is that following this AI challenge, those which, if they do meet our kind of a minimum standard, that we can work with them to improve their algorithms and maybe go on to further prospective or multi-reader studies. So we're currently in the training phase, and we hope to do the test phase come from late November onwards. So I don't have any results to share with you, unfortunately, at the moment. But just to quickly plug at the end about the importance of having a public data set, which ReIMAGINE will eventually be, just to say that NCITA is and ReIMAGINE have the PROMISE MRI data set available for access. And if you'd like to access it, you can email this email on the screen. But it's a very novel and very unique data set with a very comprehensive pathology template mapping biopsy in over 500 MRI DICOMs. So a great tool if you're developing AI in prostate. So thank you very much.
So thank you very much for the introduction, and also thank you very much to Professor Gleeson for his talk, because he's set up part of my talk very nicely. So I wanted to give you a flavour of some of the work we've been doing at Cambridge during the COVID-19 pandemic, and also try and highlight how NCITA has been really integral to facilitating our work. So going back to the start of the pandemic, the mathematics and radiology departments kind of led on forming a large multidisciplinary collaboration with partners from industry, the NHS and academia, to try and develop some artificial intelligence tools that would be open source for everyone to use, that could be deployed anywhere. And so we managed to kind of grow quite a large data set with images from across the globe for that purpose. And throughout the pandemic, we were meeting on Zoom at 5 p.m. every day to kind of push these things along. And we developed chest X-ray models for diagnosis and prognosis. We've got segmentation algorithms to show areas of COVID disease and quantify the disease. And we also developed a system to allow us to integrate with the hospital system, so that we can take images from PACS, which is, for anyone who doesn't know, the system in which images are displayed in the hospital, and then send back results, because we didn't have something for that before. So I'll give you an example of some of the work in a moment, but just wanted to highlight how NCITA has been really integral to everything we've done. So I think, like everyone, getting access to data at the beginning can be quite tricky, and convincing the hospital that we've got systems that mean that we're able to keep the data securely in a good format was really essential to give us that data access. And additionally, the data viewer allowed us to be able to review the images remotely with scrolling through CT stacks, for example, without having to download patient images to our own computers and use local DICOM viewers. So that also kind of helped with maintaining our data security, and just the way it's kind of set up meant that we were able to maintain good data management practice and store a large amount of images, as you can see. And that's just two of the data sets we've got. So one from obviously the local hospital, and then the national COVID-19 imaging data set that we also use. Now, to come on to some of the work we actually have been doing, one of the first things we did was develop a COVID-19 diagnosis tool. So this was back when PCR wasn't so readily available, and there was, it was only 80% sensitive and so on. So we used a variety of methods using different imaging resolutions and different techniques. Don't worry too much about those now. But you can see across the board, we managed to obtain fairly good accuracies of up to 90%. But I think this is where I really need to thank Professor Gleeson for his presentation, because on the right-hand side, you can see 8 chest X-rays with the saliency maps, so almost heat maps, which give us an idea of where the algorithm is looking. And a lot of the time, the algorithm appears to be making its decision either on text on the image or medical apparatus like pacemakers and lines, or even what view the image is taken from, whether it's taken from the front or the back. And so we obviously didn't feel comfortable with progressing with things like this, and we found that we did a systematic review and found that these problems are kind of endemic within the field at the moment. So what we've tried to do about it is it's a lot easier to take these things into account where there's metadata and that's accurate. So maybe that's the machine and model of the scanner, but where there are things that are either incorrect or incomplete in the metadata, whether that might be whether there's text on the image or not. We've built a pipeline that will automatically label those things, and then developers can take those into account. But that's everything. Thank you very much for listening, and look forward to answering questions in a bit.
Thank you very much for the introduction. So hello, my name is Cathal McCague, and I'm an honorary radiology registrar and PhD student based in Cambridge. And today I'm going to talk to you about our study, Integrated Image Analysis in High-Grade Serous Ovarian Cancer. Excuse me. So ovarian cancer, just as an introduction, is the 6th most commonly diagnosed cancer among women in the world today. In the UK each year, there are approximately 7,500 new cases, and unfortunately 60% of these are diagnosed at an advanced stage where the curative outcome isn't so good. So the real challenge with ovarian cancer is that we're not actually treating a single disease, we're treating, a myriad of subtly different diseases, and that's kind of the unique biology which creates these clinical challenges. So the disease itself, the name is a little bit of a misnomer because it doesn't actually originate from the ovary, but rather from the distal end of the fallopian tube. And what happens is we get the development of invasive serous carcinoma on the fallopian tube, and then carried on the peritoneal fluid, we get dissemination of cancer cells throughout the abdomen. And the challenge is that as the cancer cells locate into different disease sites, common ones being around the liver capsule, under the diaphragm and in the omentum, we get development of disease which is genomically and immunologically different from one another, so intersite and intrasite heterogeneity. And this proves a real challenge when we're trying to assess treatment response using something like RECIST 1.1, because we can get response, in one lesion and not in another disease site, and we can even get subtly different responses, so part of a lesion responding and another part not responding. So we've been trying to create something which does a better job than RECIST 1.1 essentially, and those techniques have been based on volumetric analysis of the whole disease burden, and also development of radiomic tools to increase our insight into the disease response. Those techniques both require robust and effective, in terms of usage of time, segmentation. And currently, the only way we can do this is manually, but it can take between 2 and 3 hours per patient, because the disease is so voluminous and so technically challenging to segment. So to this end, we've been trying to build algorithms to automatically segment and to radiometrically analyze these disease sites. And doing that, we've used data from the ICON-8 trial. So the ICON-8 trial, just as an introduction, was a randomized 3-arm phase 3 trial aiming to understand the safety and efficacy of dose-dense, dose-fractionated carboplatin paclitaxel chemotherapy in the treatment of these patients. And it first recruited patients in 2011 and has just recently stopped recruiting patients. And for our study, we've tried to recruit all centers with more than 15 patients. So here are the 24 which we've been in contact with, and our study started in early 2020, and obviously has been curtailed massively by the pandemic as resources have been diverted away from research. So I am happy to say, however, we've been able to recruit 15 centres, spreading geographically all the way from Leeds in the north down to Somerset in the south, and have almost recruited 500 patients. So these 15 sites were those with most patients, so we've targeted those specifically. And for each patient, we have between 2 and 6 imaging time points. So approximately 1,400 CT images, which over the next while we're going to segment and then use to develop our automated segmentation and radiomic tools. And our project aims to create an all-in-one cancer imaging pipeline, which goes from automated reconstruction at the start through to segmentation, radiomic analysis, and then developing multimodal treatment prediction tools, which can allow us to deliver personalized treatment. And the NCITA repository has been totally fundamental in this, because it's been the place where our 15 centres have been able to access remotely and then upload the scans using software created by Lorena Escudero Sanchez, which automates the anonymization and upload process. So it's been really super. The work that I'm doing focuses on the automated segmentation, and I'm just going to mention one project which we've done to this end. So we've created a tool which can automatically segment ovarian cancer on CT scan, and this is to speed up the process, which I said is manually very time consuming. And the outcome in terms of dice score, created a tool which had modest dice scores, I would say. And we wanted to understand if they actually translated to scans which were noticeably different from ones that an expert radiologist would produce. So we ran a visual Turing test where we had 20 testers from a spectrum of expertise, all the way from genitourinary radiologists at the top in terms of expertise, all the way down to medical students. And we asked them to look at 80 CT scans and asked them whether the segmentations had been created by a human or not. And we found actually that there was no statistical difference in classification accuracy between the experienced groups. So having expertise in ovarian cancer actually didn't translate to knowing whether these scans were segmented by a machine or not. So that's led us to try to develop tools which better assess segmentation quality. And we're working on developing a new metric, different to DICE, which will actually encapsulate clinical utility, having a radiologist at the center of the process. And for that, we're very grateful for the NCITA's support. So thank you very much for your time. And I'd like to just highlight the work of the group in general. So the radiomics and quantitative imaging group who've all collaborated on this project. Thank you.